Welcome to the next episode of the Microbiology Institute. So today we'll be talking about the DNA sequencing by the Sanger method. So what is the DNA sequencing? So as we know, there are four bases in the DNA. So that is the adenine, guanine, thymine and the cytosine. So the process of determining the order of the bases in a sections of the DNA is called the DNA sequencing. So DNA sequencing is used for the identifications of bacteria, is used for the identifications of the genome, is used for determining the mutations. So DNA sequence is very much useful in today's microbiology as well as in the sections of the molecular biology. So Sanger method was described by the Frederick Sanger in 1977, for which he won the Nobel Prize in 1980. So this method is also called the chain termination method or dideoxy method. So in the figure you can see there are two types of the molecules. One is the dideoxynucleotide and next is the deoxynucleotide. So in the deoxynucleotide there is the OH in the 3' end, which is highlighted, and in the dideoxynucleotide there is H in the 3' end. So you can see there is the H in the 3' end and OH in the 3' end. So that makes it different. So all these structures are similar, but only there is the H in the dideoxynucleotide, whereas in the deoxynucleotide there is the presence of the OH in the 3' end. So what actually happens is that, so in a state of DNTP, DDNTP can bind in the DNA. So in a state of the DNTPs, DDNTP can bind. If the DDNTP binds in a synthesis reactions of the DNA, then the synthesis of DNA will stop. And these are the regions or the basis of the chain termination method through which we can determine the gene sequence. So in the synthesis reactions, if the dideoxynucleotide is added instead of the normal deoxynucleotide, this synthesis stops at that point because the 3' OS necessary for the additions of next nucleotide is absent on the dideoxynucleotide. So there won't be the additions of the further nucleotide if the DDNTPs are added into the DNA. For example, if this is the DNA and there is the adding, suppose this is the DNTP, so there will be the suppose DNTPs, and if by any chance if DDNTP get integrated, so what happens, that synthesis reaction, DNA synthesis reactions will stop here. So you will stop here, so only these short fragments of the DNA can be formed. So this is the basis of the Sanger method for DNA sequencing. So first is what we do is that the sequence of the double-strand DNA molecule is determined by enzymatic process of complementary polynucleotide chains. These chains terminating at a specific nucleotide positions, and these are separated by the gel electrophoresis, and last we read the sequence. So I will further clarify the procedures in these principles. So the requirement is the single stranded DNA molecules, then after DNA polymerase primers, 4 DNTPs, that is the ATGC, and then after small amounts of the 4 DNTPs, that is DDATP, DDCTP, DDTTP, DDGTP. So these are required. So these are the terminators. So DDNTPs are the terminators. So in the figure you can see this the double stranded DNA. This double stranded DNA are converted into the single stranded DNA. So you can see this is the single stranded DNA and there is the additions of the primer. So after the conversion of the single stranded DNA from the double stranded DNA, so there is the additions of the primer. Then after we take the 4 tubes, in the 4 tubes we add the DNA to be sequenced along with the primer. Then after we add the DNA polymerase, and the DNTPs. So after the additions of the DNA primer, DNA polymerase and DNTPs, what we do is that we add DDNTPs. In the first tube we will add the dideoxy ATP, so DD-ATP. In the second we will add the dideoxy-TTP. In the third we will add the dideoxy-CTP, and the fourth we will add the dideoxy-GTP. So in this way what we will do, we will add the DD-ATP, DD-TTP, DD-CTP, DD-GTP in the different test tube. So what will this do? This will halt the synthesis of the DNA in the different sites. For example, if you take the examples of this gene. So for example, if you see here, for example, if this is the work done by the DD-TTP, so this is done by DD-TTP. so what can be this? T can be the complementary copy of the A. So there are 1A, 2A, 3A, so 3A and the 4A. It means in the DD-TTP vessels, for example, in this one, there will be the formations of 1, 2, 3 and the 4 types of the different fragments. 4 types of the different fragments will be seen. First, DD-TTP will be attached to the adenine, 1 so the reaction will halt and only one nucleotide will be added. In the second, what will be added is that the A have the complementary copy of the DD-TTP. So secondly, another fragments will be this, and last fragments will be this. So in this way, if you consider the DD-TTP vessels, so in this vessels there can be the 4 fragments. If we consider that example only, so I am giving the examples. So here you can see the 4 fragments. Similarly, 4 fragments are obtained from the DD-TTP tubes. But if you see here, so there is also the T1, 2, 3, 4, 5 TTP, so 5T. So it means that in the dideoxy ATP there will be the 5 different fragments. Similarly, in the DD-CTP, so C will have the affinity with the G. So it means how many affinity will it have? So 1, 2, 3, 4. So it will have the 4 affinity, 4 fragments. So similarly it will have the 4 fragments. Similarly, in the different tubes there are the different fragments of the DNA. So what actually we do is that we will put that different fragments of the DNA into the gel electrophoresis. First we have the first well, which will consist of the DD-ATP, DD-CTP, DD-TTP and DD-GTP. So we have the different well for the different tubes in the gel electrophoresis. Then after we put this here, this here, this here and this here, so we leave it for the gel electrophoresis. So what happens is that on the basis of the size, what happens is that the DNA will run. So if the DNA has the smaller fragments, so it will travel farthest. So this DNA, so here you can see this DNA has the longest one. So you can see here, so this DNA has the last DNA has the longest one. So similarly you can see here, this has the second one, this has the third, this has the third one, this is the fourth, this is the fifth, sixth, seventh, eighth, ninth. So in that way we can obtain the size. So after, you can see what we have to do is that we have to read the sequence. So here example is the G. First is the G, and then after the G there is the A, and after the A C, then after there is the G. So in this way we have to know. For example, here is the first, here is the G, so there is written G. Then after there is the A, there is written A. Then after there is the C, then there is the C. So in this way we will determine the sequence, and this is the complementary copy. So what we will do, we will make the complementary copy, and actually this is the sequence of the DNA. So this is, in this way we can determine the sequence of the DNA by the Sanger method. So thank you for watching my video. So if you really like the video, please don't forget to subscribe my channel and don't forget to share this video. Thank you.